Is it turning on? I'm now paranoid that the projector never works. Okay, I know you had a chemistry test. Both of you, not all of you. I know that it's Friday. I know that you have break. Oh, we're still gonna talk about children. That too. I spent a little bit too much time on this. That's true. I love it. Silly fish. I think we're not sleep, release. Question mark?
Don't think I do. Sometimes I can answer that question, yes.
Sometimes I haven't too.
I don't think I have one in this bag.
Okay, it's like stuff.
One time I was making up with my daughter. But we have this warm back home. I'm not upset with you, I'm not consume you, I just can't.
Thank you. I normally have some of this.
I do in my bag.
Okay, one more minute.
Yeah, I'm taking all my freshman class. You know, that's.
Okay. The video stopped, so we're gonna go over this. Peripheral, Nadaria, or both? Diploblastic. Both. Amoebocytes. We're good on the first one. Peripheral. Medusa. Nadaria. Melanocytes. Peripheral. Asexual reproduction. Both can do it some. Radial symmetry. Area only gets nutrients via diffusion. Both of them. And true tissues. Area only. So I want to clarify, I said on Wednesday that true tissues meant that they had to have like a specific function, but they also, I read more about it, I want to make sure I correct what I'm saying. They have to be separated from other tissues by a number of layers. And so that's another reason that peripheral don't have true tissues, they don't have a separation of layers, one layer from another layer. Okay, what is A in the picture of this peripheral? What is A? Starts with an O. Starts with an O. The opening to the outside. The osculum, yes. What is B? This is what I threw in later. I remember saying, oh, I didn't say this right. No, that's a development. I mean, it's kind of but not really. That's in a more in an area. It's just the spongiocele. It's like their version of a seal. And see what kind of cell is shown in C. And actually, it's kind of two cells there. The spongiocele is B. And then C, I acted like that's one type, but now I'm looking at the picture better, and it's really two. I was thinking more of the one that has the flagella and the collar. That's a choanocyte. The blue one is an amoebocyte, but they're both in the same box, I'm noticing. Those little yellow things, remember, are the spicules that help provide structure. Here are my answers. Oh, they're there. I know I have answers. So today we're going to talk about Nadaria and a couple other ones. I'm going to give you, this is the plan as I see it for now. So I was informed that there was not a link for the chapter 32 outline because there wasn't one. So there is one now, but I feel kind of guilty. So I've changed the due date for it right now. If you're already done, just turn it in. But I changed the due date to the 28th because I'm not gonna make something so great, but I told you verbally it's instead of I did not put that on rules anyway, so. Chapter 32 outline then, for chapter 33 outline, it's gonna be due that following Monday, which is the 6th or 7th of March. So it'll be March already. Or I might do it Wednesday, I don't know. It'll be due not the week to get back to the week after that. Chapter 33 is a monster. It's a really good chapter. We're gonna finish chapter 33 the week after February break. So the current plan, I think, looking at our schedule, your next task is on 31 through 33. And I think it makes more sense if I just finish 33 with you and write the test because I've been here for the whole thing, rather than having her take over in the middle of the chapter and write a new test. Write a test based on what she wasn't here for or what I wasn't here for. So I think I'm going to teach Monday, Wednesday, Friday when we get back. And your test is going to be that following Monday, which is the 7th, I think. It's the 7th. So you can write that down and I'll put on a little sometime for you. Okay, March 7th, Monday, March 7th is going to be your test on chapters 31 to 33. And your outline will either be due that day for 33, will either do that day or Wednesday. Okay. Questions on the schedule? Okay, I don't think of any problem getting through 33, but then I looked at it, I only got through 2 slides for 33 the other day. So we'll see. We'll get through it. Apparently, I'm more passionate about invertebrates than I realized, is what I realized as I was looking through stuff. So we started talking on Wednesday about Nadaria. And I honestly don't remember what I said and didn't say. I know we talked about that it has true tissues. We talked about that it has radial symmetry. It's diploblastic. I know I used the term nerve net because I referenced my friend Paul. I don't think we talked about reproduction really at all, right? Am I remembering right? I don't think we did because I don't have anything scribbled up here about it. So we talked about the fact that there's 2 body plans. There's a polyp
which is a sessile attached to something with tentacles up, kind of. And then there's a medusa form with a tentacles down that's mobile, or at least can sort of move itself, or is controlled by the currents. I told my kids I thought we should do invertebrate charades. And then I was like, but pretty much everything would be like, or for now. So I was like, we can't do charades. So we talked about eating, the fact that they use their tentacles to bring food in, that they're carnivorous, that this yellow is kind of their gastrovascular cavity. So they bring things in, they put enzymes in there, release enzymes into the water that's in there. And that helps to digest stuff. And then they just, because they're thin enough, it can diffuse through the cells. We didn't talk about the center layer, I think, between the two layers. So this yellow is the endoderm. This blue is ectoderm because they're diploblastic. They only have those two layers. This mesoglia is just similar to what the mesohyl is in the periphery. It's just a gelatinous layer in between the two, the lining and the outside layer of the organism. Again, I don't think we talked about reproduction at all, and we hadn't talked about stinging cells at all, right? Okay, that's where I thought we were. I just wanted to make sure. Okay, so I read a book a couple summers ago, not last summer, the summer before, about jellyfish. If you want to learn more about jellyfish. So I struggled with what to read to you because I would like to read you the whole book, but we don't have time for me to read you a whole book. So this section that I'm going to read you talks about the jellyfish life cycle and reproduction. And here's a diagram that I don't think is in your PowerPoint because I pulled it up when I was looking at this. So this woman was an ocean scientist and she spent a lot of time, she quit her job to take care of her family and then started writing a book about jellyfish and traveled all over the world learning about jellyfish. And there's a lot of interesting ecological stuff about jellyfish, but they're just also very interesting biologically, and we're still learning a lot about that. So this is a section in the middle of a chapter where she was talking about this guy who was trying to make commercially viable jellyfish tanks that he could sell to people so people could have jellyfish sitting on their desks. So he was trying to understand how to reproduce jellyfish because he needed to make jellyfish to sell the jellyfish. So understanding what it takes to breed jellyfish requires a little jellyfish sex ed. So is jellyfish sex any good? Certainly there's no tedious dating. Jellyfish don't waste many or any energy on seduction and allure. But as far as interesting sex, jellyfish have that in spades. Here's how it works. A medusa, which is either a male or a female, produces either eggs or sperm. Some burp up their genetic material, cast it out into the wide open ocean, and hope for the best. In other species, a mother jelly receives a packet of sperm from a male and broods her young among her tentacles. Either way, if a jellyfish sperm meets a jellyfish egg in the same species, new life begins. But now the jellyfish life cycle veers away from the familiar. A fertilized egg grows into a larva called a planula. That one jellyfish aptly described as looking like a furry tic tac. The fur is a rug of eyelash like extensions called cilia. Once a newly minted larva has its cilia wiggling, it starts swimming. Upon bumping into a rock or a dock or a boat hull or an alive petri dish, it performs a spiral dance that ends when it attaches itself to the surface. And then, like an enchanted creature, it begins shape-shifting. The larva elongates into a tube and grows wispy tentacles. It builds a digestive tract down the middle of the tube and a mouth in the middle of the tentacles. This being is now called a polyp. So she's focusing on scyphozoa, which are the jellies that swim around. So the polyp is down there. No, the polyp is there. Sorry, it's called a scyphistoma. It's in the middle. Sorry, I forgot all of them. Unlike human infancy, the polyp is not a brief stage that the jellyfish passes through. A jellyfish polyp is a fully realized organism. The jellyfish can be a polyp for much longer than is the more familiar medusa. The length of time varies for different species. Some jellyfish probably remain polyps for years or even decades. Alex pointed out in his breeding tank next to the greasels, which held dozens of fuzzy petri dishes sitting in still seawater. It was not the kind of fuzz Alex wanted. What he hoped for was the fuzz of moon jellyfish polyps. What he had was a coating of algae. Alex wanted moon jellyfish polyps to grow in these dishes as a way of getting a large quantity of medusa. He needed to stock his tanks. Once you have polyps, they make more of themselves, explained Alex. While the medusa reproduce sexually, the polyps reproduce asexually, and they do it very well. One polyp can multiply itself to make new polyps, sometimes fields of them, sort of like the way you shoot some grass will proliferate in the dark. But that's not all. Some polyps scoot along the seafloor and leave behind the most extraordinary footprints. A piece of the polyp's foot breaks off and remains where the animal had been standing. This dollop of polyp. This dollop of polyp forms a tough coating around itself, of which part is called a protocyst. When I first heard the word, I thought of it as a seed pod, but I later realized that the code comes from the Greek word for foot. Still, a seed pod isn't a bad mental image. The protocyst can lie dormant for years until one day the conditions change and it sprouts into a polyp, much like a plant sprouting from a seed. As if the shape-shifting, cloning, and forming foot seeds weren't enough, the next stage in the jellyfish's reproductive process is perhaps the most bizarre. At a cue from the environment, like changing water temperature or salinity, the polyp's stringy tentacles are absorbed into its body. The tube grows even longer, and what looks like a stack of pancakes form where the tentacles used to be. The name for this process is strobylation. Forked branches extend from the center of each pancake, forming a delicate snowflake-like organism. The pancakes begin to wriggle individually. The forks flicker, and with what is sometimes described as a pop, the top pancake flutters away from the stack. Now the next pancake in the stack intensifies its quivering, its forked branches extend, and it too flows away. This continues down the stack. Thus freed from its sessile existence, the baby medusa is called a phyra. You can see that there, which looks like a stack of stuff in the middle, and then that phyra pops off. A name shared with an ancient Greek sea nymph who lived in a silver cave in the depths of the Aegean Sea. The phyra come from hardy sea stock. The phyra came from hardy sea stock. She was one of 3,000 daughters of Oceanus, the god of the world oceans, and Tethys, the sea goddess. So too, this newly budded Aphyra has abundant siblings. Depending on the species and the polyp's health, between 1 and 60 Aphyra bud from a single polyp. The budded Aphyra pumps its short arms a few times and swims into the sea. It begins to eat tiny plankton and soon grows to about 1/3 of an inch across. Its bell inflates, it grows tentacles. Oral arms extend from its mouth. If there's food available, the maturing Aphyra is capable of growing an inch every few days. Soon the creature is recognizable as a medusa, and it's likely seen clustered with hundreds or even thousands or millions of creatures with very similar DNA. Other medusa from the same polyps and its clones. Once strobilated, the polyp doesn't just roll over and die. Polyps have the potential to continue to clone and strobilate over and over again. That's why Alex needed to grow polyps. Without polyps, he'd never get medusa, and without medusa, he wouldn't have a disease. I thought that was a slightly more dynamic description of the life cycle than just pointing at the picture and saying, okay, it becomes a planula, and then it can become this schistosoma, and then the phyra break off. They're weird. They have a weird life cycle, and that's just the jellies. Again, we didn't talk about all of them. We're going to talk about their classes, but before we do that, we're going to talk about their stinging cells. So this is a picture from your book. One thing that makes Cnidaria is their specialized cells. You can see here's a tentacle, and that could be a tentacle of a polyp or medusa. And as you zoom in, there's two specialized kinds of cells that you can see here. You've got nematocysts and you've got cnidocytes. So cnidocytes are the stinging cells. That have the toxin in them. That they use to inject the toxin into something that they are stinging. Okay, what's kind of interesting about these is that they're single use only. So every time they use one, they have to make a new one. So it's a pretty intense process, so they're not just stinging casually. And these are unique to cnidarians. They're the only ones that have them. That nido comes from nide, which is Greek for nettle. I don't know if anybody's ever been stung by a stinging nettle plant. It's memorable. It happened to me probably 10 years ago for the first time. I was walking through these plants and all of a sudden I was like, ouch. I was like, these must be stinging nettles. I've never seen them before. I didn't know what I was doing. Has anyone been stung by a jellyfish? It's also memorable. And not in a good way necessarily. Thankfully, you were not stung by some of the species of jellyfish that could kill you. That would not be as memorable for you. Within the cnidocyte, then there are these specialized nematocysts. These have like you can see here, a coiled like thread. Ooh, that's supposed to be say threaded. A coiled thread with a barb on the end. It's actually barbed throughout. So this is what causes the pain. It's a poison and a actual physical barbed thread going into your skin. See if I've yet mastered playing a video. Ooh, maybe I have. Except there's no sound, so no, I haven't. Is this muted here? Okay, it must be muted here. No. What in the world? This is on. Oh, nope, it's not muted here. Every time I think I'm gonna get this, I don't. If I skip the ads, you think it's gonna work? Escape it and what? Oh, this, oh, a good call.
Tingling sets in.
Okay, we gotta go back 1 second. I do know about that setting. I just assume it's already on for
You're
this.
swimming in the ocean and something brushes your leg. When the tingling sets in, you realize you've been stung by a jellyfish. How do these beautiful gelatinous creatures pack such a painful punch? Jellyfish are soft because they are 95% water and are mostly made of a translucent gel-like substance called mesoglea. With such delicate bodies, they rely on thousands of venom-containing stinging cells called cnidocytes for protection and prey capture. Even baby jellyfish, the size of a pencil eraser, have the ability to sting. Larval jellyfish, ephyrae, look like tiny flowers pulsating in the sea. As they grow, they become umbrella-shaped with a bell at the top and descending tentacles around the margin. The largest species of jellyfish, the lion's mane, has tentacles that can extend more than 100 feet, longer than a blue whale. These tentacles contain most of the stinging cells, although some species have them on their bells too. Venom is ejected via a nematocyst, a whip-like hollow tubule, which lies coiled under high osmotic pressure. When mechanical or chemical stimuli activate an external trigger, the lid of the cell pops open and seawater rushes in. This forces a microscopic barbed harpoon to shoot out, penetrate and inject venom into its victim. Nematocyst discharge can occur in less than a millionth of a second, making it one of nature's fastest biomechanical processes. Nematocysts can continue to fire even after a jellyfish has died, so it's important to remove lingering tentacles stuck to the skin. Rinsing with vinegar will usually render undischarged nematocysts inactive. Seawater can also help remove residual nematocysts, but don't use fresh water, because any change in salt balance alters the osmotic pressure outside of the cnidocyte and will trigger the nematocyst to fire. That's why urinating on the affected area, a common folk remedy, may do more harm than good, depending on the composition of the urine. Most jellyfish stings are a painful nuisance, but some can be deadly. An Indo-Pacific box jelly, also called a sea wasp, releases venom which can cause contraction of the heart muscles and rapid death in large doses. There's an antivenom, but the venom is fast-acting, so you need immediate medical intervention. Despite the impressive power in their tentacles, jellies aren't invincible. Their stinging cells are no match for the armor of thick-skinned predators like the leatherback turtle and ocean sunfish. These predators both have adaptations that prevent slippery jellyfish from escaping after they are engulfed. Backwards pointing spines in the turtle's mouth and esophagus, and recurved teeth behind the sunfish's cheeks. Even tiny slipper lobster larvae can cling to the bell of a jellyfish and hitch a ride, snacking on the jelly while they preserve their own energy for growth. Small agile fish use the jellies as moving reefs for protection, darting between tentacles without ever touching them. Nudibranchs, which are sea slugs covered in protective slime, can actually steal the jelly's defenses by eating the cnidocytes and transferring them to specialized sacs for later use as weapons against their own predators. Even humans might benefit from the sting of a jellyfish one day. Scientists are working on manipulating cnidocytes to deliver medicine with nematocysts rarely 3% of the size of a typical syringe needle. So the next time you're out in the ocean, be careful, but also take a second to marvel at its wonders.
Are you marveling at its wonders? Okay, we're gonna talk about a couple of the classes within the phylum of the Cnidaria. So there's two major clades within Cnidaria. The Medusazoans are ones that obviously have a medusa form. Nope, stop, I'm not chatting with anyone. And these are your, again, the jellies. Remember, we're not going to call them jellyfish. And we use that term, which I'm going to check my spelling of again. Scyphozoans. They have both tentacles and oral arms. So the oral arms are what brings stuff into the mouth. So they have specialized tentacles almost that can bring things into the mouth. They also have a contractile ring. So remember we said that animals, muscle tissue was unique to animals. These guys don't really have muscles, but they have bundles of microfilaments that allow them to contract, and that allows them to control their movement. Their GI cavity is basically acting like a hydrostatic skeleton for them. So they fill their cavity with water, and then they can contract against the pressure of the water to be able to move themselves through the water. The sea wasp they referenced in the video as being a very deadly one, and it is. That's from a class of cubozoans. These guys don't have oral arms. They only have tentacles. And they have 4 corners, which we can't really see on here. So their bell kind of has 4 corners called a pedalium. And that's where their tentacles are attached. So they have a different arrangement of those tentacles. They're really fast jellyfish, and they're really deadly jellyfish. Okay, these are the scary ones. These are the ones that some of them can kill you in 3 minutes if you get enough poison in you. They're also really interesting because they actually have eyes. Like cornea, retina, lens. It's not a really great eye that they can make out strong forms, but significant. They're able to differentiate between light and dark well. There was a movie that came out many years ago involving jellyfish and Will Smith. I don't know if anyone saw it. Yes. 7 pounds. Yes, I had to look it up. It was a traumatizing movie for me. I was extremely traumatized for it, but it involved a box jellyfish. That's what I remember about it. I just remember it being traumatic, and I didn't like it. And I don't think the premise of the movie is well founded, but there's a box jellyfish in it. I'll just say that. I don't want to ruin it for anyone who wants to watch it.
We all thought you were talking about Shark Tales.
I also. think it's our-
Is Will Smith in Shark Tales?
Yeah.
Is he a voice in that?
Yeah, it's awesome.
I haven't seen Shark Tales. I'm sorry. I'll put it on the list. I'll add it to the list.
Very, very good.
Okay, that came out after I was at the animated movie age, I'm guessing.
So.
No, I'm just saying, I watched all the animated movies as a child and that would have come out after that period. And then I didn't have children yet, so I hadn't gotten to, I just watched Cars for the first time, like 3 weeks ago, because it didn't, I was past that point in my life. I'm sorry. I liked it. Okay, so Shark Tales, I'll write it down. My son loves sharks. There's a shark in it. And apparently there's jellyfish in it.
Yes.
Okay, I'm sorry. I was thinking of a more dramatic version of a Will Smith movie that involves jellyfish. Apparently he likes jellyfish and he's in a lot of movies about them. That's another picture of a scyphozoan. Okay, we gotta come back to this. Okay, also under the Medusazoans are, and I don't know, I don't have a picture of them. And now this picture's in my way. Yes, I want to write. I'm going to pick orange. This picture is not a hydrozoan. I just need space to write. Okay, hydrozoans are another class. They actually alternate between, they have a polyp, and orange was not the right color. And your book goes through the life cycle of Obelia. We will look at that one in lab the week you get back. It does some asexual reproduction via budding, but it can also reproduce sexually. The second group then, second clade within here, within this, are the anthozoans. Anthozoan literally means flower animal. And you can see why they get that name, because they look like little flowers, but they're animals. They only have a polyp form. These are corals and anemones, so some of them, some of the corals, I shouldn't say all of them, can secrete calcium carbonate. That's a 3. It doesn't want to be a 3. Why are we writing in dots now? Calcium carbonate. So they can make their own exoskeleton of calcium carbonate. And a lot of them live symbiotically with other organisms, which you talked about when we talked about like dinoflagellates and algae that live within Cnidaria to make corals. Okay, it's a mutualistic symbiotic relationship where everybody benefits. And when conditions get bad in coral reefs, they will actually release, the Cnidaria will release their symbionts, they'll release their algae or dinoflagellates, and that's what's called coral bleaching, which happens for a variety of reasons and may or may not be happening more regularly depending on who we talk to. Okay, that's Cnidaria. We've gotten through 2. Look at us. Only 3 more to go. Only these are like really big ones that have lots in them. So that's the last of the ones that are not bilaterally symmetric. Okay, so everything that we talk about from here on out, you're not writing, has bilateral symmetry. And they're all triploblastic. So they all have a mesoderm during their developmental process. Most of these that we're talking about also, and again, remember I'm saying most, it's like the protist again. Most of them have a digestive tract that has 2 openings. And most of them have a true coelom of the ones that we are still going to talk about. So we're gonna start with a Lophotrochozoa. Thankfully, your test will not be a spelling test. Okay, Lophotrochozoa have two, and when I say have two, I mean most of them have one, and some of them have both. They have a Lophophore, which I think we've mentioned before, right? Or is it just that I've read about it like so many times that I can't remember? This is a crown of ciliated tentacles. Not tentacles, sorry. Yes, I guess it is called tentacles, not. It's a crown of ciliated tentacles that are used for feeding. So that's where I'm like, oh, this is all we've got for charades right now. Lophophore. Polyp. It's all the same. The other thing that some of these have, some of them have both of them. Some of them only have one, is a trochophore, which is how we get the name Lophotrochozoan. Lophotrochozoan. Luckily, it's not a spelling test for me either. That's why I have my notes here. And this is a larval stage that is free swimming. And it has cilia. So some Lophotrochozoa have both the Lophophore and a trochophore. Some have one or the other. And some of them are molecularly related and don't have either. So we're going to talk about 6 different phyla that are within the clade Leucotrophozoans. Not today. We're not going to get through 6 today. We will get through one, maybe not even one. We'll see. Free swimming with cilia. I do not resent any questions about my handwriting, which is only made worse by this situation. Okay, so Lophotrochozoans, we're starting with the phylum Platyhelminthes. So Platyhelminth is Greek. I think I did not write that down. I don't know if it's Greek or Latin. I think it's Greek. Meaning flat worm. These are flat worms. I realize I lack any kind of flatworm clothing. It's a hole in my wardrobe I need to fill. Okay, so not surprisingly, flatworms are flat. They have flat, long bodies. Again, worm is kind of a generic term, just kind of referencing long bodies. There's a lot of different kinds of worms, and we'll spend a lot of time talking about worms. Okay, so remember, we have those 5 questions we're thinking about for all of our organisms. We already talked about the fact also that these have bilateral symmetry because they're in this clade, all of them are bilateral. So they have bilateral symmetry. They're triploblastic. These do not have a true coelom. Most Leucotrophozoans do, but they are pseudocoelomates. What does that mean? That's the word we used and defined, but it was a couple days ago. No, because they are triploblastic. Yeah, so the mesoderm doesn't fully, like they don't have a coelom really, but their mesoderm, they sort of have this. Now wait, stop for a second. I read about too many last night. These are not pseudocoelomates. These are acoelomates. I'm like thinking about it in my head and looking at the picture in my head and thinking, no, they're acoelomates. They don't have a coelom. What? Yeah, so they have no body cavity. They're really thin bodies, which means they're like the other organisms going to do a lot of things by diffusion, but we're going to see some start of specialization in terms of having something. So we had those 5 questions. How do they eat? How do they get rid of waste? That kind of thing. So when we talk about them eating, they have a branched gastrovascular cavity, which we'll see. Forgot those were there. We'll see in a minute. So they have a branched gastrovascular cavity, but they're still doing most of their movement around their body of nutrients is still by diffusion. So they have this branched cavity so that they can get the nutrients close enough to every part of their body. But the nutrients themselves are still moved throughout the body via diffusion because they don't have any kind of like circulatory system or anything like that. But the branching of the gastrovascular cavity lets the nutrients get close enough to all the cells in the body so they can just use diffusion. They also exchange gases. By diffusion. Again, because all their cells are close enough to the external environment, they don't need specialized organs for this. They're living in moist environments. A lot of them are aquatic. Some of them live in the soil. But they're living in a moist environment. They need that moisture to be able to do diffusion. And their flat body gives them better surface area for diffusion. Remember the whole surface area to volume ratio. They're thin, but they have a lot of surface area because they're flat. And this we're starting to see in terms of getting rid of waste. We're starting to see specialization. They have little cells, little clusters of cells called protonephridia. Protonephridia. So protonephridia is referring to like kidneys. Proto is like before kidneys. So this is like a precursor to kidneys. These are helping to get rid of or help control the osmotic balance in their bodies. So they're simple excretory organs. Your kidneys do a lot to control the osmotic balance, the level of water and salts in your body. That's what these are doing. And they have these things called flame bulbs or flame cells, which have cilia in them that beat to draw water through the tubules. So they help move the water through the organism. I tried to find a really good video of this and I did not. There was like one 12-second video on YouTube that looked like a flame cell out of context, so it wasn't a great video. There's lots of animations of it, but I couldn't find an actual real video of it. So these help to control osmotic balance, but they're getting rid of most of their nitrogenous waste still via diffusion. So they don't have like a really developed excretory system with bladders and stuff like that. Again, because they have a simple flat body, diffusion can do a lot of what they need to get done. They can range dramatically in size. Some of them are microscopic. Some of them can be 20 meters long. And we'll talk a little bit more about that in more detail than maybe you want to. When we come back from break, one of the things that we'll do for lab is we will have planaria. What's really cool about planaria is they have the ability to regenerate. And so this leads us into our reproduction. You can take a planaria and cut it in half and it will grow a new head and a new tail and be 2 planaria. And you're going to try that. You're going to try to chop little planaria in half carefully so that you can have more little planaria. So obviously they can do asexual reproduction.
How big are they? Are they like microscopic or?
The ones that we're going to work with, The ones that we'll work with are not microscopic. They're probably 2 centimeters, a centimeter and a half to 2 centimeters. Depends on how big they are when they send them to us. Probably 2 to 3 centimeters would be maximum length. So they can regenerate. Some of them also reproduce asexually via budding, which just means growing a whole new worm off their end of themselves. And it's asexual because they're growing an identical copy of themselves. And some of them do just kind of split in half. Fission. Like the planaria did with binary fission. Not planaria, paramecium. We're talking about planaria. You can see on this one, I think they're cute. I like to watch them. I just wrote over it, but they have eye spots, which are sensitive to light. Again, it's not like they could come in here and get a great picture of the room, but they can sense light versus darkness with their little eye spots. And not only can they do asexual reproduction, they can also reproduce sexually. And again, most of them are hermaphroditic. We talked about the peripher being sequentially hermaphroditic. These guys are not sequentially hermaphroditic. They're usually simultaneously hermaphroditic. So they have both male and female sex organs. And so they can reproduce by mating and they just switch sperm. So they cross fertilize by each of them giving sperm to the other one. So within the Platyhelminthes, before we go on, I don't have a video, but I want to show you this picture. They just discovered 2 new species of flatworms.
I saw that.
And they found them and they're outside what they think is their native range. So they think that people have introduced them, but this is Humbertium covidum, named after COVID. Partially, I liked this because it's an homage to the numerous casualties of the pandemic. I was like, would you want a flatworm named after you if you died from COVID? I don't know. But also because a lot of the study that they wrote about it was written during the pandemic when everyone was locked down. I thought that was maybe a more apt reason to name it that. But I think he's kind of cute. He's a terrestrial flatworm. And they call him a hammerhead flatworm. I don't think there's any more pictures. I think that's the only picture. Yeah. So when we talk about things like classification changing and flatworms, maybe you don't get excited about it, but we're still discovering lots of new things about animals. So they also have some beginning of cephalization. Oh, no, don't do that. Oh. Look at that great picture. I need to erase all my writing now. Nope, go back. Remember how I was going to master this? Still not done it. No. Where did you go? Down here. Okay, fine. There we go. They do have the beginnings of cephalization because again, they have those eye spots and there's a concentration. That's a concentration of sensory gathering information at one end. So they have a distinct head. And this is just showing you the flame cells. Remember those protonephridia? You've got the flame cell with the cilia that are beating, and that's just moving stuff through. And then they have these excretory pores to get things out. So again, very, very primitive nephron kind of structure, which you'll talk about kidneys later in the semester. But the same kind of idea. I would love to say that we were going to go on and talk about trematodes, but it's too complicated for the next 1 minute that I have. So have a great break. Watch 7 Towns to be traumatized. Read Spineless to learn more about jellyfish.